And you can, okay, maybe it's something with my microphone. Maybe I need it on.
Okay.
Okay, I'm good?
Yes.
All right. Well, thank you very much for inviting me for this evening for you, for morning for me, to talk about sterile environments and cleaning validations, inspections, warning letters and trends with the US FDA. So I just wanted to go over just some of the uh, I go over the FDA website and I see what's current and what's going on with the US FDA. And so this presentation is just meant to kind of summarize that, just to get an idea of what's going on in the US and how that may impact Singapore. And it's just kind of a fun little exercise to look and see what's going on with that group. So the goal of this talk is really to understand the FDA and understand their expectations. And by understanding their expectations, you're able to better defend your own process. So if you know what it is that they're going to ask about when they come to your facility, you can be better prepared. Now, that's not saying that you should have a good quality systems in place for their visit and approach it holistically, but you should be able to at least get an idea of what they're concentrating on and what they might look at when they come to your site. So how we got here is I reviewed the FDA observational findings to date, as discussed in the previous slide, and then just kind of put my own current industry perspective on it. So this isn't an FDA endorsed talk by any means. It's just looking at their data, looking at what I think it means and summarizing it up. And also just giving you some tips on how to shore up your own cleaning validation program and your own critical environment control program. So what I did is I polled a bunch of my colleagues out there. It wasn't a poll in a presentation or any poll like that. I contacted people who I know what their role was in industry, and I reached out to them and I gave them a list of questions. So one of the questions that I wanted to know, because I've been really out of an FDA inspection other than being on the phone with a client or working through their observations with our customers, I wanted to know what were some of the most typical observations or focus areas with respect to cleaning validation that they are seeing in the industry over the past year. So I contacted some of my friends and colleagues that I knew were in recent inspections and just kind of picked their brain and asked about what they were seeing. So the answers were the basics. So a lot of times when they were being audited, FDA was really covering the basics, show me your master validation plan, show me, walk me through a protocol. How do you qualify or, validate this piece of equipment? So it was really the basics. What they also said was that most programs, and this may be from some of the consultants, do not hold up to a cursory review, much less an in-depth one. So there were some issues, and we'll see that when I show you the trends for the 483 observations and I rank them. I show you the top 12. And you can see how some of the most basic things are ending up as 483s. So, just not having the proper documentation in place. A lot of times we go through the exercise, we have a meeting and we talk about it, and then you deal with it, but there was no documentation of that process. So you need to document what you're doing, and even if it's a simple risk assessment on how you choose a sampling site, that needs to be documented. Trend for inspectors wanting to expand the scope of CV cleaning validation. And I've seen this a lot. I hear this a lot from from some of our customers that we work with, and when I benchmark other people's programs against other people's, that the auditors or inspectors are trying to expand the scope of cleaning validation to, the outside of the equipment, or, to disinfectants, or, they're just trying to keep expanding that scope. But, your cleaning validation program should consist of, what I call your product contact parts or your wetted parts, and maybe some indirect surfaces as well, like a buffer formulation tank is an indirect surface, and inside of a lyophilizer, things like that. So, you want to make sure that you define what your scope is for your cleaning validation program in your master validation plan, so that you have documentation for what you decided was in your cleaning validation box, so to speak. Inspections are becoming more focused on data integrity than any of the science or studies. Seems to be the buzz these days, so that there is a trend for, looking at data integrity, looking at your recipes and your programs on your equipment, looking at audit trails, making sure that each operator has their own password or their own login and their own sign-on. So, just making sure that your data integrity program holds up. Observation tied to equipment, exteriors, rooms, and floors. So this kind of goes with that other bullet point of trends for wanting to expand the scope of cleaning validation. So again, observation tied to the exteriors, rooms, and floors. I would probably push back on that. Insufficient testing to support carryover limits. So again, I see this a lot, that there's not a lot of testing to support the carryover limits. So you want to look at your design space. You want to make sure that you have that data in your life cycle approach. You want to make sure that you're getting that data in your first phase. Data integrity issues again came back up. So what we're going to do with the remainder of this talk is we're going to talk about what the FDA expects, discuss some current cleaning validation trends, and discuss current critical or sterile environment trends. So starting out, what does the FDA expect? So I went all the way back to 1993 to the cleaning validation guidance document. And this document was a guidance document that was intended for the FDA inspectors on how to inspect cleaning validation programs. So it was never really meant for us, it's meant for them. But if they have a guidance that's telling them what to look at, we should understand that document and know that we're going to get inspected on those parts. We're going to get inspected on that document, so that we should understand it as well and use it as a reference. So, even though the document is, over 25 years old, it still is a good document that you should review and at least have an understanding of that document. So from that document, they want documented evidence that an approved cleaning procedure will consistently reduce active pharmaceutical ingredients, process residues, cleaning agents, and microbial residues from product contact equipment surfaces to acceptable levels for the processing of drug products. So that's right out of the cleaning validation guidance. And then to summarize it, there's other guidances out there also that you may find useful. So, these again, they're good documents to refer to, even though they're a little bit, they're a little older right now, but there's, the validation of cleaning processes, which we mentioned before. So let me see if I can find my pointer, my laser pointer. So there's cleaning processes. You can look at the high purity water systems, parenteral lyophilization, pharmaceutical, micro quality control labs, dosage form drug manufacturer GMPs, OSDs, sterile drug substances. So, all these are very good guidance documents that you can also refer to. But getting back to the validation processes 1993 document, what it does or what they expect. So this is basically the document outline. So I went in, read it, outlined the important parts of it. So here it is. They want written procedures detailing the cleaning process. So what that means is that you will have, written procedures that just don't say, they want a cleaning process that has basically similar documentation as a batch record. So I've seen cleaning documentation or cleaning SOPs that are, super complicated. And I have seen some that just say, disassemble the piece of equipment, pour the detergent in, and clean the equipment. Check. Um, so you have one that's deficient because you should have sign-offs, you should have important parameters defined, and you should have design space in there where you should have ranges for your targets. So you want to make sure that you have written procedures. What that scope in that written procedure and how detailed it is, that's up to you, but they should at least pass a regulatory scrutiny. You want cleaning processes, cleaning process residues removed. So you want to make sure that you are removing whatever it is that you're using to clean your equipment. So if that's a detergent, you need to make sure that those residues are also removed. If you're using solvent, same thing. You want to make sure that those are removed. You want to have written validation procedures to address who is responsible. So you want to make sure that you indicate, who's going to be performing the validation, who's going to be testing, who's going to be approving. You want to have acceptance criteria in there, and that should have been defined earlier in your life cycle. So you shouldn't just get up to your validation process and then have some acceptance criteria there that you pulled out of the air and that you don't know if you'll be able to meet them. And then you want to make sure that you have a revalidation timeline or a way to assess that continued process verification. You want to make sure that your validation protocols address sampling procedures and analytical methods. And you want to make sure that you follow the protocols and document the results so that you're accruing that data and then wrap it all up in a final validation report. And I have at the last bullet point, I have ×3, because do we really need to do it 3 times? 1 time is an isolated incident, 2 times is a coincidence, 3 times is a trend, right? That's what we say in validation. But if you have enough data moving forward, maybe you can only do 1 time. 1 time might be sufficient. But if you have something that's seasonal, let's say something where I'm recommending more than 3 is if you are using maybe a biologic that has some impact of raw materials, or maybe you have a botanical that is impacted by raw materials, or maybe you have something seasonal that is going on. So you want to look at, not just 3 times, but maybe you need to do 10 times to assess the different raw materials and seasons, for example. So in 1993, that guidance document was drafted, but another thing that happened in the US with respect to the US FDA was the Barr decision in 1993. So both things happened at the same time, and obviously one came after the other. But the FDA initiated an action under the Federal Food and Drug Cosmetic Act to be able to take items off the market. So before this Barr decision, so quick, let me just go over some of the things that happened with this. So with this court decision, what it did is it strengthened the FDA's regulatory power. And there were some definite deficiencies with this company. So they needed to produce a study to validate that the firm's regular cleaning procedures actually clean its milling machine, identify the cleaning agents used in all its cleaning validation studies, and failure to validate test methods and manufacturing processes, including the cleaning process. So this actually came from a judge. So a judge in the United States court was telling the pharmaceutical industry, hey, you guys need to do this. You need to validate that your cleaning process actually cleans. I mean, it seems so simple, but it actually took this Barr decision to kind of bring it to the forefront in 1993. So it's something that really wasn't being done before that. But it also strengthened their power by being able to demand that a company recalls their own products. Because in the past, to take items off the market, the FDA either had to have the firm involuntary, they actually had to have them voluntary recall, or they had to request a court order to seize the products. So, now it's like, okay, well, if the company has products out there that are deficient, it's like, okay, can you please take them off the market if you could find them, or they had to go to court and get a seizure. So what this did is now this set a precedent so that in this decision, the FDA can actually ask the court to order the responsible firm to recall the products. So basically, the judge ruled that the FDA requires companies to perform cleaning validation, is what this decision really meant. So how important is a robust cleaning program? So we know that that Barr decision was a big court decision, really bolstering that FDA regulatory power. But what it really means to a company is that, let's look at what drives companies. Companies like to make money, right? So a robust cleaning program is really important so that your company can keep making money, keep making their products. So I have an example here of a firm called KV. And this was a firm that this statement right here, upper left-hand corner, KV was deficient in keeping conditions sanitary. Their cleaning practices were terrible. Things weren't cleaned properly, including the machines. This is from the FDA audit. I've never seen in an FDA audit where the FDA inspector actually says their cleaning practices were terrible. Usually it's they assess something specifically, and they'll say specifically the equipment wasn't clean per blah blah blah, 21167A. But they were just overwhelmed with this. So what happened was that federal agents seized more than $24 million in unapproved drugs on July 30th of 2008. So the U.S. Marshals um, actually seized the product. The seizure followed an inspection of several companies' plants where an FDA investigator found the company was not complying with FDA enforcement notice. So what happened here is there was not even a program to run FDA audits. FDA came on site, and they were on site a good 6 to 7 hours before somebody from quality was notified even that the FDA was on site. They were back in the lunchroom, they were talking to people, they went in the restrooms, they just walked around and found everything, uncovered a lot of things. But what this meant for the company is that they had, so you have July, you have that inspection that happened and those seizures that happened, and they had a consent decree later of that year, and then you see the stock tumbled, and the company became, I don't want to say worthless, but it definitely a stock hit, and it impacted the company, lots of layoffs. And so what happened, why I am intimately familiar with this, is because I was hired here to try to help the company come back up to an acceptable level of quality. So needless to say, upper management didn't embrace this. They continued to try to make drugs and keep doing their same practices, and the company ended up going bankrupt and folding. So what that meant is company lost a lot of money, shareholders lost a lot of money, and a lot of people lost their jobs. And really, a cleaning program is vital to keep a company operational, because you can make all the product in the world, but if you can't do it in clean equipment, you're just going to end up recalling your product. So why is this so important? Why do auditors come to a site and start asking about your cleaning program? So why they do it is that the cleaning program really shows an auditor a lot about your facility. So it shows that whole quality mindset. How does your site make decisions under duress? When you are trying to get product out the door, are you cutting corners? Are you skipping important steps? Are you testing into compliance? Are you blaming an operator for, if you have a corrective action and you're constantly, calling it training, it's a training issue. If you have 10 training issues and you have something that's going on with your process and you keep blaming the operator, you don't have a robust CAPA system, you don't have a robust auditing system to make sure that your CAPAs are meaningful and that they are actually working, and you don't understand your process. So there's a lot. So just with that whole quality mindset. It also shows how a firm will investigate deviations. So if your firm is, again, constantly not implementing meaningful corrective actions that actually correct the problem and prevent the problem, it shows you don't understand your process. So some of those observations that show this is, for example, your firm does not have data to demonstrate your cleaning processes for non-dedicated manufacturing equipment and utensils are adequate. So that's just an example of when I see something like that, it just shows that facility doesn't have a good handle on their cleaning validation program. So moving in, we have warning letters and Form 483 observations are available to the public. So how I went through and got some of this data is I went to this reading room, and I've provided the link up here, and then the data dashboard down here. So you will have my presentation. You can, I don't think it'll work if you click on the link, but you can copy this into your browser, and you can get the data dashboard, or you can look and look at some of those 483 observations. But I did it for you. So why do the FDA make this data available? So first of all, they believe that the information will help the industry improve quality. So by sharing those common observations, you know what the FDA is concentrating on, but you can also learn from others' mistakes. Learn before it impacts you. It identifies possible areas of emerging concern. So an example of this is in the US, there was, I'm sure this went all over the world, but there was the New England compounding pharmacy issue. And what happened is that they had contaminated steroid injection that they were injecting basically into people's spinal fluid, and it was killing people. So what happened is the FDA did the inspection. They found out that this compounding site was actually doing manufacturing, but they were not following any of the manufacturing protocols, and they were not following the GMPs because they were calling it specific to a patient, that they were compounding the product for a patient, but they were compounding it in huge batches. So because of that, the FDA started inspecting a lot of these compounding facilities. And so when you went back in time, if you go back and look around maybe 7 years ago, you could start seeing the trend of sterile, of manufacturing to provide a sterile environment. You can start seeing that 483 observation pop up all over the place. So you can start seeing that, oh, okay, they're looking at aseptic procedures right now. They're making sure that your micro programs are in place. So all of those 483 observations started bubbling up to be in the top 10 because that's where they were focusing. Now the FDA is focusing a lot of their efforts on breakthrough drugs, a lot of COVID right now. They're focusing on a lot of the still compounding facilities. So you can start seeing where they start showing their concern. A lot of data integrity, a lot of documentation issues. So what this does is it helps you avoid warning letters by avoiding the 483s in the first place and having your program stand up to the scrutiny of a regulatory inspection. So looking at the total number of inspections, now this is just drug manufacturing facilities. So the FDA inspects food facilities, they inspect a lot of different facilities. But what I did is I just, veterinary medicine, things like that. So what I did is I just focused on the drug manufacturing facilities. So the FDA inspections of foreign drug manufacturing facilities has increased after 2006, so that's something that you can see. So the blue line is the domestic inspections that have been formed just for drug manufacturing facilities, and the red line are foreign inspections, so that you can see they're kind of coming into an equilibrium that are doing both. And this is really the globalization of the FDA drug inspection program. So in response to move from domestic to global manufacturing and the passage of FDASIA, which is the FDA's drug inspection program, it shifted from one that was heavily focused in the US, so back, in 2000, all the way up to about 2006, about 9, 10, right around here. It shifted since 2015, really. It's conducted more foreign than domestic drug inspections. The FDA drug inspection program is now risk-based. The FDA will prioritize inspections facilities deemed higher risk based on scientific defined criteria. So what that means is more foreign inspections. So what they use is a site selection model. So to address the need to prioritize use of limited resources, in 2005, the FDA implemented this risk-based uh, this risk-based approach to drug facility surveillance inspections. So what they use as a mathematical model was designed to select facilities with the greatest potential for public health risks that and ones that may not comply with established manufacturing quality standards, new facilities not yet inspected, and those having complex manufacturing steps. And what this does is it results in the model to prepare a prioritized list of facilities for inspection. So it was really that FDASIA, the Food and Drug Administration Safety and Innovation Act, that was passed in 2012. So what this did is this act supported the risk-based approach, removed the requirement to inspect domestic facilities based on a fixed biannual schedule. The FDA now has been able to inspect some facilities that previously had not been inspected. And CEDAR's catalog showed that of July 2016, there were 965 foreign manufacturing facilities that have never been inspected by the FDA. By the end of last year, they had inspected 495 of those facilities, or 51% of those facilities that have never been inspected. So it also requires facilities to provide records in lieu of or in advance of an inspection. So now it gave them authority to deem these drugs adulterated if foreign facilities delayed, denied, or refused to permit an inspection. So it really gave them a little bit more power globally. So before, the Food and Drug Cosmetic Act required that biennial inspection of drug and biological products, but since FDASIA was passed in 2012, the biennial inspection requirement was really replaced with this risk base. So that just means more foreign inspections. It's going to level out, which the data does show that it's leveling out between domestic and global, and that they can request those records in advance and really kind of perform that inspection almost remotely or ahead of time. So as far as FDA enforcement actions, they had, so what there's Form 483's inspectional observations. So these are when they're performing an inspection, they come on your site, they see a deficiency, they will issue a Form 483 inspection. Sometimes these can even be remedied before they leave the site. So if it's something small like this SOP doesn't have a secondary signature for critical steps, we can go ahead and revise that SOP if you can push it through your system quick enough and revise that while they're still on site and show them, and they may just remove that 483 observation since you fixed it real time. So it may be something super simple that you can fix right away. Warning letters, but again, Form 483s, most will not be able to be remediated. Sometimes it's, your quality systems are deficient or your documentation is not there, and it may take a little bit more time. So when you are dealing with the FDA and you are getting those 483s, you want to make sure that you're just not agreeing to everything. You do want to push back a little bit if it's something that you know your process the best, and if it's something that you documented and that you thought about, you did a risk assessment, and you have what you think is a solid program, you can say, that's a nice, if they tell you to do something, and again, once it's in a 43, you're committed. But while you're talking to them and while you're in your audit, you can ask them to explain things. You can also explain your stance on it and say that, that's a nice, thank you for providing that feedback. This is the way we do it. We feel that, we understand our process, and just explain what you're doing. And sometimes that's good enough for them if you can explain it and why you're doing something. So warning letters in 2020, there were 168 issued, product recalls, 145, seizures, there were 3 seizures and 12 injunctions. And I do not have a number on consent decrees and civil prosecution, but you can go to this website and you can for yourself and look all of this data. So again, this is the example that I was talking about before, where the pharmacist that was linked to the New England Compounding Center. And the pharmacist actually was arrested because the indictment charged that the supervisory pharmacist told technicians that production was more important than cleaning and disinfection. So what happened in that, there were 751 patients that were actually diagnosed, and this was across 23 states in the US. The 751 patients were diagnosed with this fungal infection. And so it was prednisone or injectable steroid, and 64 of those patients did die. So anyway, that's, I just wanted to kind of highlight that. And then you can see some of that data shift from 2012 to now because they're focusing more on sterile environments. Okay, so moving into inspection references. So inspection references are, it's information provided to FDA investigators and inspectors to assist them in their daily activities. So I provided the link here that you can go in and you can look at some of these, these inspection references, and in it you'll find compliance manuals. So there's a compliance program guidance manual, or a CPGM. These are instructions to FDA personnel for evaluation of industry compliance. You will find compliance policy guides, which is policy and regulatory guidance for FDA staff. You will find inspections operation manuals, or IOMs. And this is a procedure manual for FDA personnel performing inspections. And then you'll also a manual on foreign inspections. So again, here is the link specifically for the foreign inspections so that you can take a look at what they're doing. And then all the manual, the link is up here. So looking at, I want to focus now on the Compliance Program Guidance Manual, or the CPGMs, that provide the instructions to FDA personnel for evaluation of compliance. So in it, you can see over here on the right, of some of those programs. Highlighted 7356.002, the Drug Quality Assurance Program. So what it is there's a set of 6 systems to inspect. So there's the quality system, facility and equipment systems, materials, production, packaging, labeling, and laboratory. So these are the systems that touch, they come to your site for an inspection. And it's right there in their CPGM. So you can look at this and see what they're going to look at. So as far as pre-inspection planning, the Office of Surveillance will prepare and get an up-to-date site dossier consisting of quality information on inspection history, recalls, shortages, customer complaints, foreign inspection outcomes. Remember, we have the agreement now, and all the agencies will share data. Any submitted quality metrics data, so they'll look at your annual product review. They'll look at any kind of changes out there that are in industry. And then they'll have a list of all products manufactured at the site. So they'll look at they'll focus on low dose products, narrow therapeutic range drugs, combo products, modified release products. If you have any ER or extended release products, they'll take a look at that. Biologics and new products. So this is kind of where the focus as far as products and in that risk assessment. So that when they come to your site, they already have all this information that they requested, and that they have this dossier consisting of all this information that will help them really, by the time they get to your site, they're already formulating what they're going to start looking at. So the Investigations Operations Manual, the IOM, again, it's the procedure for FDA personnel performing inspections. Here is the link to download the one that's pertaining to equipment, but I that out for you. So in section 4.5.4.5, equipment and utensils, here it states, and I highlighted, I thought this pretty interesting, arrive before processing begins. Evaluate conditions and practices not as observable before plant startup. Because a lot of times what we want to do is we want to have the facility running. We want to just walk them by a set of windows and have them look in, but they want to get in there. They want to see what's happening before. They want to assess the cleanup, where equipment is stored, how equipment is stored. Are personnel sanitizing their hands and equipment before beginning their work? So they're looking at all of things. They want to look at these things. They want to examine all of the equipment and utensils, determine the design, materials of construction, the maintenance of the equipment, suitability, the ease of cleaning. They want to open inspection ports, notice if inspection ports have been painted over or sealed, and determine if the equipment is clean prior to each use and the method of cleaning. So they are looking at their your cleaning program. You know, in it, it's a bulleted list of what they need to look at. So I encourage you to look at the performing inspections, this IOM, and to look at the utensil section that's in there. As far as FDA trends in 2019, so I can't look at it, all the trends, I can't look at it to date because it's, you know, the data is uploaded and it's on a rolling year. So I can only look at it for a certain period of time. So I looked at it on a 12-month rolling period up to, believe it was October of 2019. So that's the most recent data that I can trend, at least for the purposes of this talk. So the regulations haven't changed as far as our Code of Federal Regulations. It's still the same regulations. You know, they're very vague. They don't tell you how to do things. They just tell you what's expected. But they use a computerized system that allows more risk-based, better focused inspections based on these industry trends. So they're looking at these industry trends and it's kind of feeding upon itself. So if they're seeing a lot of issues with, you know, equipment cleaning, then they're going to focus more on that, and then they might find more on equipment cleaning. So it's, you know, it kind of rolls that way, but it's what it is. The top citation that's out there is procedures not in writing or fully followed. So the top citation so the first, second, third, and fourth all pertain to procedures, controls, and investigations. So we're not documenting our processes very well. We're not investigating sufficiently. So those are some things that the FDA is focusing on. Procedures for sterile drug product, again, it increased sharply. So from 2018, it was number 12. From number 8 with 62 citations in 2017 and 79 in 2019. So it is increasing years of sterile drug products. So not only did it increase, you know, 8 years ago when they started focusing on that, now, you know, since those feed into their risk base, they're focusing on it more, and those may be causing more and uncovering more. So 67A, so part 211, 67A, equipment and utensils not cleaned, maintained, it moved from number 12 in 2017 to number 5. So now that's a dramatic increase. They're focusing on cleaning equipment and utensils. They are focusing on that regulation right now. And the difference between a regulation and a guidance is the FDA telling you, this is what we like to do. It's a good idea. Do you have to do it? It's an expectation. So I always say it's an expectation that I expect when I make dinner, I expect that my husband cleans up, right? Or that he takes out the garbage. He doesn't have to do it, but there's going to be a consequence if he doesn't, right? So that's kind of what the FDA guidances are. They're expectations. So they're not laws, but you better abide by them or you're going to make them mad, right? They're going to start digging if you're not following the basic guidelines. Now, with the regulations, those are laws. Those are, you know, you need to follow these. So looking at the trends overall, you see that the top FDA Form 43 observations. So I have, let's see, 1, 2, 3, 4, 5. I have the top 12 listed out here, I think. 1, 2, 3, 4, 5, 6. I never know how many I put. Sometimes I have 10, sometimes I have 12, but I think I went back 12 for you guys. So what I see here, you have the light blue line is 2016, then it goes 2017, and then 2018, and then 2019. This is the most current data. so that you can see procedures not in writing, it's increasing, but also, you know, the number of inspections are changing. So they're hitting different facilities, they're hitting new facilities, so they're uncovering all of these things. Investigations of discrepancies is number 2. Scientifically sound laboratories controls is number 3. And written procedures is number 4. So remember, 1 to 4, controls, investigations, and documentation, all 1 through 4. And then you have number 5, equipment and utensils shall be cleaned. And I think that these 4 that I highlighted relate to cleaning validation programs. So you can see here in 2016, it was stable, 2016, and then it kind of went down, and then it spiked. So then we're at 81 and 99. So this moved, it was out of the top 10. Now it's number 5. So expect your equipment and utensils to be, the cleaning programs to be inspected. Procedures to prevent microcontamination. Again, little increase here. So it went from 48 in 2016, 62 observations, 50 to 79 observations in 2019. And then you see equipment design is following the same trend exactly as equipment and utensils. So even it dips and then goes up. So they're looking at how your design, the materials of construction, can it be cleaned? Are you cleaning? Are you documenting your cleaning? All of these are what they're focusing on. And then procedures for cleaning equipment. So procedures are kind of steady. So they're just kind of hanging in their steady state. So let's talk about understanding your process. So they're focusing on cleaning programs because what they want to do, or what they want to know is, do you understand your cleaning process? So I have an example here of a site. It's a case history of a site that was cleaning a bioreactor. So they have a bioreactor, and the bioreactor runs for 14 days. They use antifoam. Okay. The bioreactor currently takes 8 hours to clean. They use sodium hydroxide and 10% citric acid. The tank is not visually clean after processing, and the residue is removed by wiping. However, not to confined space concerns. So what happens is, with this piece of equipment, they were getting in, they were cleaning it, and then they were going in with a ladder, manually cleaning, and as far as they can. So the entry can damage the bioreactor, obviously. The process time is confusing, and this would require a total of 3 people to clean. So it is an option, although they had to do it at some time to get it clean. So they wanted to fix this. So to understand your process, what we recommend is, you do your coupon testing or your evaluation and your design when you're designing your cleaning process. So what we did is we coated stainless steel coupons. We did a coupon study with them. We looked at their cell culture residue for 14 days. We heated it and then air dried at ambient temperature for 16 hours. And what we discovered is that the cleaning agent, it took 1% formulated alkaline detergent to clean, 25 degrees C, and 15 minutes. So that's a whole lot better than 8 hours and manually scrubbing using the sodium hydroxide and 10% citric acid.
Hi.
So you have to know that, yes.
Your slides are frozen. If you can just.
Uh-oh.
Yeah, we're still.
Are they still?
Trends.
Oh, on trends. Okay. Let me share again. Can you see something different now?
No.
No?
No. Do you want to, I'll stop this.
Let me stop sharing and then come back in.
Yeah.
Mm-hmm.
You got my chat for some reason.
Mm-hmm. Well, hang on.
Yeah.
Can you see a new slide now?
Yes, cleaning process design.
Perfect. Okay, thank you. All right. So with your cleaning process, when you have an issue and you're not able to clean your reactor in this case, you need to look at everything. You need to look at your design of your equipment. Can it be cleaned? Did you do your riboflavin coverage study? Does it have the right flow, the right pressure, coverage, everything? Look at your materials of construction. Look at the soil. What is your soil that you're trying to remove? How much is on the surface? What are your manufacturing conditions? Are you heating it? Are you baking it on? Are you hitting it with another chemical that's going to make it harder to clean? What cleaning methods do you have? Your mechanisms that you would require to clean that chemical? What are your cleaning parameters? So what temperature do you have available? What are you using to clean? What concentration of detergent are you using? What kind of detergent are you using? Or what kind of chemical are you using? And your cleaning agents. So it really, you want to look at everything. So you want to look at the soil, you want to look at the surface, you want to look at the cleaner. So we started asking them some questions about their process, because it's like, okay, well, we're able to clean it relatively easy. They were having issues when they tried to implement our recommendation at site. So we started talking to them about, are you cleaning stainless steel? We found out that, the design of the equipment was fine. The materials of construction were fine. But then when we started asking about the process a little bit more, we found out that they were using something called Petamac, or polydiol dimethyl ammonium chloride. So it's what it is a charged polymer, and it causes your cells to basically precipitate. So your solution becomes easier to filter. So what happened is, so what it does, you put it in your solution and it causes the precipitation. So it clarifies your product so that you can either, go on to your next step. So whatever that is, but most likely it's purification. Filtration. Sorry, maybe I should have had that cup of coffee. So anyway, so what it is, it's this charged polymer. It is a it's a polycationic agent, and it binds to the negatively charged cells and the cell debris. So it will form a clump, clarify your product. So what happens is we talked about, flocculation and anti-foam with the customer, and, that they were using, they had 20 million viable cells per mil density, and then implementing the Petamac, or that charged polymer. And what we noticed was that it actually made the product harder to clean. So we went back, did another coupon study using the Petamac this time, emulating their process. And what they found is that when they implemented the new cleaning process, they were able to save, have about a 25% reduction in their chemicals that they were using. They didn't have to enter their tank anymore, so they didn't have to go in with 3 people, do lockout, takeout, go in and into confined space to manually clean this tank. They saved 29% time, so it was quicker, and they calculated that out to a savings of about 85%, or 2.9, or sorry, 2,905 euros per cleaning. So that's quite a big change, just knowing how what's in your process, designing your cleaning program to adequately clean your process. So it really, you have to understand what's going on. So that's what we determined that they needed. So that's a little bit different from the 1%. So they required 2% formulated alkaline detergent plus 2% oxidative detergent. So let's move into calculation of acceptance limits and some of the changes that are going on there to help you shore up your cleaning program. So current situation right now, there's a little bit of confusion out there right now with setting limits. So the EMA published their guidance on health-based exposure limits, or HBELs as people refer to them, on the 1st of June 2015. And then Q&A followed that in 2018, so April 19th. So this is really the timeline for limits. So there's that PDA Guide to Inspections that happened in 1993, And then you had the Foreman and Mullen paper determining cleaning validation acceptance limits in 1993. And then a PDA Tech Report 29 points to consider for cleaning validation in 1998 that was revised in 2012. So really there was a void here. So everybody, you know, we had that 1993 document, we had the 1998 document, and then we were just chugging along. Everybody's doing the same thing. And then in 2010, ISPE came out with a baseline guide for risk-based manufacture of pharmaceutical products, or risk map. And this really kind of introduced the ADE approach, or using health-based exposure limits to calculate your acceptance criteria. So before that, we were, you know, using a dose-based MACO calculation. We were using therapeutic dose, 1/1000th of therapeutic dose. So there was other ways to calculate that, you know, defaulting to 10 ppm at the dose base or the MACO calculation. So then what happened is the EMA implemented the guidance on setting health-based limits in 2014, made it effective in 2015. So it was really in their guidance at that point. And then the effective document in 2015 for existing products, and new products in December of 2015. And then they published the questions and answers in 2018, which kind of confused people a little bit more. So we're going to talk about that. So industry perspective. So again, I asked my colleagues and friends out there, if acceptance limits were discussed, what was the stance of the EMA, FDA, et cetera? So any inspector agency that came into their facility, and acceptance limits were discussed, what was their stance? So did they require calculation of PDE and establishing a lower process capability number or alert action type of limit? What did they want? Because we know that the ISPE never really intended for the ADE to become a limit, right? It's supposed to be a level of risk. So there needs to be some amount of control within that level of risk that you're controlling your process. So there was just some confusion in the industry. So what do you do? Do you do capability? Do you take, you know, have alert action limits? Do you call it a more, a par? What do you do? So some of the responses I got back, so I love these. Agency personnel, for the most part, do not understand the PDE concept either. So we're all learning together, right? Agency fine seeing multiple limits, then using the lowest, also fine with process capability. So right now, you know, they're fine with seeing multiple limits, but they're also fine with seeing capability. Alert limits were discussed with an emphasis on lowering these if the data supported. Discussion on what actions would need to be addressed if a trend of alerts were noticed, since they are not technically action limits that were exceeded. So there was some pushback. Sorry, I want to go backwards. I'm going forward and I want to go back. So this one I thought was interesting because this came out of an inspection from one of my friends. And so the FDA demanded that they implement alert and action limits. And so they were like, they pushed back. They said, show me, what do I use? Do I use 80%? Do I use 50%? I don't have any guidance on what to use. So we're going to keep doing what we're doing. And they did not get a 483 for that because they're really, you know, there is no guidance on that right now on what to use if you're using alert and action limits. But it's a good idea to have something. So you don't want to make them mad. You want to work with them. So prior to this, in 2014, people were using PDA Tech Reports 29 and 49. And in it, you know, as discussed, we talk about minimum therapeutic dose, or 1/1000th of a dose, using toxicity, or LD50, converted to an ADI, an acceptable daily, and then ADE, and then a threshold of toxicological concern, or a TTC. All these were referenced in PDA Tech Report 29, which was to consider for CV, and it's a 2012 revision. So the first document was in the 90s, second document was in 2012. And so this paradigm shift happened where the ISPE document that we discussed that came out in 2010 really established those health-based limits, or ADEs. And what this was is a true margin to say, so if your acceptance limit is up here, And this is your true, this is really where your true residue is. This is your margin of safety. And then the 2014 guidance on health-based limits, it really solidified that risk map approach. Get some water. And in the 2015 document, qualification and validation, section 10 cleaning validation, it really mandated HBELs for the EU. So it becomes a thing then in 2015. PICS and WHO followed suit. So PICS has a document that they, couple of documents that became effective in June 2020 this year, inspection of HBEL assessments and use in quality risk management. So this is an aid memoir. The purpose of the aid memoir is really to define an approach that inspectors can consider when inspecting HBELs. Any HBEL document that they have, it prompts to consider expectations, and it has PICS references that are used that are recorded, as well as references to the Q&A document here. So this Q&A on implementation of risk-based prevention of cross-contamination in production and guidance on setting HBELs for use and risk identification. So the aid memoir can be used as a supporting document for inspection of cross-contamination risks in shared facilities. And then the WHO had a draft document May 2020 that they talk about points to consider on different approaches, including HBELs to establish carryover limits for cleaning validation. So the calculation of the ADE, I just wanted to go over so everybody knows what I'm talking about. So the ADE is your no observed adverse effect level multiplied by your body weight divided by a bunch of modifying factors and uncertainty factors and pharmacokinetic adjustments. So the ADE would take the place of nature in that limit calculation, and there would be no other safety factors applied in that original MACO calculation. PDE is very similar. It's still using your NOAL and a weight adjustment, but then it has a set of values that are used to extrapolate between species, variability between individuals, repeated doses, severe toxicity, and if a no effect level was not established. So they'll score it. So this F1 will be scored between 2 and 12. So depending on what it is, this factor of 10 between individuals. So it's going to look at just differences between, what the study looked at. This F3, or let's just talk about F1 between species. So F1 would be, you would score it a 5 if it was a rat to a human, or a 12 if it was a mouse to a human, or 3 for a monkey to a human. So it would be scored that way. So F3, then, you'd score it for 1 if it was greater than half a lifetime study. It would be 2 for a 6-month rodent or a 3.5-year non-rodent study. So it's scored that way, and those are your safety factors. So as far as calculation of a safety threshold value, or an STV, this is out of the ISPE document, you would take your ADE, multiply it by your batch size, divided by your maximum daily dose, and that's now your safe threshold value. So again, the safety factor is already in there. You don't need to apply anymore. So, this is your traditional MACO calculation. And this is what I was talking about, having a process capability. This would be your process capability right here, your CPK. So you could also define them as process control limits, or what I like to call a manufacturing operating range, and then a proven acceptable range. So you could call this your PAR, this your MORE, or you could use the critical limit where you can alert and action limits. So I see people do everything really, but I think industry is moving more towards a process control limit. And so what you would do is you would take your CPK and your upper specification limit, subtract your average of your measurement divided by 3 standard deviation to come up with a capability. Now, per the original ISPE baseline guide, your risk map, it states in there, ADE should not be seen as a limit. It's an initial acceptance criteria. That's a limit that's safe for patient. Use it as a reference point for determining your level of risk, and then establish your process control limits. So really the ISPE document, if everybody would go back, and refer to that one, that's really what we should be doing. The EMA, I think everyone's kind of coming to that agreement now, but right out of the ISPE document, it says ADE should not be seen in a limit in a true sense, but as a reference point for determining your level of risk. So again, you may want to establish mores and pars, an inner control limit, set that tighter than the out of spec limit, so that when you're exceeded, then it could just be a document, it could be it could just be a note in a batch record or a comment in a batch record rather than a full-on investigation and taking that batch out of service. So setting limits, my advice is to determine your PDE value. This becomes your PAR. Collect your data for that predetermined amount of time. So you want to describe it. You want to have this up. You want to have this written in your cleaning validation program. Use data to set your manufacturing operating range or your process capability limit, and have a plan in place for when your MOR is exceeded. So example, investigate and comment for an isolated incident or a formal investigation into trending the data, whatever. But you need to have a plan in place for when your inner limit is exceeded, because again, you have all that level of risk, but there's something going on with your process that made it go out of trend. So again, some have set percentages and assigned alert and action limits. I see this. I saw this a lot at first, but it's kind of trending down, where others, because they're pushing back on the FDA, but they're really moving into the MORs, PARs, and process capability. So quickly, let's talk about trends. I don't have my watch on, so okay, so I have some time to talk about that. Pardon?
You have 20 minutes to include questions also, so.
Okay, so let me just do a couple minutes just talking about Annex 1 and some of the changes, and then we can take questions. How about that?
Thank you.
Because this is important for your global audience. So draft Annex 1, and it finally, I say it finally arrives, but almost. The update was announced in January of 2015, and the reasons for the revision were really to the ICH Q9 and Q10 not included in the 2007 version. There had been advances in sterile manufacturing since 2007. There's been a little bit of ambiguity in the document, in the 27 document, and Annex 1 scope to encompass use beyond sterile manufacturing. So they really wanted to revise this document. It has a lot of input. It's been going on for about 5 years now. It's a draft right now, so these may change, they may be changing, but I just wanted to go over some of the highlights. So again, it's not a revision, it's a rewrite. There are 269 clauses compared with 127 in the 2007 version, and 100 new clauses with no direct link to an existing clause, so it's completely new material. When the update was announced in 2015 concept paper, there were, we were told of those reasons, but really, it's a rewrite in true sense of the term. So there are 14 clauses from a previous revision not included in this new revision, and 70 clauses from the previous revision, which may have some impact on manufacturers. So new topics include process water systems. So again, this may have changed since the last time I've looked at the draft, but there was a requirement to sample from the worst case sample point each time water is used for manufacturing. I'm not sure that's possible. So hopefully that has been revised. I looked at my most recent draft and it was still like this. So it may, again, these are just word of caution. Take a look at these when the document becomes effective. There are new topics with respect to operator qualification. There are new single-use technologies, so interaction between product and surface. So what this is, leadability and extractability testing. Of course, I know there's been a lot of industry, I don't want to call it, but a lot of industry dialogue, I'll say, on PUPSIT, so pre-use integrity testing now, and compromised packaging. So again, this is one that will, a lot of people talking about it last summer and even into the end of 2019. There are new topics on critical utilities. enclosed manufacturing systems. So with respect to sterility, integrity, your background environment, and material airlocks. So this one I thought was interesting. Material transferred into an area should be on an authorized list. If not on the list, it should be an exception. So this means everything that you're taking in the facility, you should have an authorized list. Now, my thoughts on that, not that anybody cares, but my thoughts on that is that you should have at least something that worked well in a facility I worked at is we had an environmental review board. And so that environmental review board really looked at anything that you were bringing into the facility, whether it was a cleaning, something to clean a tank with, or a sponge, or gowning material, or the pens that you're bringing in to a piece of equipment. And they really kind of approved everything that was brought in was a critical change, but we didn't have an authorized list. And this authorized list, it should be an exception, because what if you I don't know, I just see this maybe not being so discrete, but maybe have a little bit of fluidity to it. But I just thought that was one that was interesting, that materials transferred into an area should be on an authorized list. It provides greater detail, almost too much, for clean room classification. So a requirement for higher numbers of sample and volume for aseptic processing, even talking about what to do in case of missing stoppers, even down to clean room clothing type, talking about dedicated socks before being into a grade C and B. So they're really getting into the weeds here on this document. So that's why there's so much, back and forth in industry with this document right now. So as far as disinfection, the differences between the documents. So right now, the 2007 version is in yellow. So where they talk about where disinfectants are used, more than one type should be employed. So there was a little bit of ambiguity there. They clarified that by stating more than one type of disinfecting agent should be employed, should include the periodic use of a sporicidal agent. So they're clarifying what they intend, but there's still a little ambiguity in there, because the industry has really moved away from using rotation of two disinfectants, where they consider rotation to be a sporicidal agent and a disinfecting agent. But with this wording here, it makes it seem like they're still requiring more than one type of disinfecting agent. So still a little bit of ambiguity in there, or not. Maybe that's what they mean, the intent. So here they're talking about monitoring. It should be undertaken regularly in order to show the effectiveness of your disinfectant program. And they're talking about removal of disinfectant residues in your cleaning program. And the old document, or 2007 document, said monitoring should be undertaken regularly in order to detect the development of resistant strains. So this is improvement, because we know that in a pharmaceutical environment or a manufacturing environment, there really isn't going to be resistant strains. That's really kind of been disproven. So rotation should include either two disinfectants in sequence with a regular rotation with a sporicidal agent, or one disinfectant and a sporicidal, rotated in there. That's really what it's meant, but in industry is kind of moving from the disinfectant model. So with that, I think I have about 15 minutes for questions. So, in the last slides, I just talk about how come you don't really develop resistance to disinfectants in a clean room, because it's not the same as an antibiotic. It's really not going after a cell wall or DNA or anything. What it's doing is it's basically annihilating that microorganism, and it doesn't have a chance to recover from that. So a quat would literally poke a hole with the surfactant, and the cell would leak and eventually die. So there really isn't a resistance in clean rooms. So I do want to open it up for questions now, and I guess I can get rid of this. So you will have my slides, you can look at that, and we have a couple of book chapters out that discuss, for example, this study right here talking about residue removal. And one thing I do want to point out is that there's a ISPE cleaning validation life cycle applications, methods, and controls, which they alluded to that I was on a committee. So this is the most recent document that came out. It was effective, or I guess it came out in September of 2020. So there's a link to that document if you'd like to purchase it. It's a very good how to perform a cleaning validation. So there's a lot of good how-tos and tips in that document.
Thank you very much.
So we can take some questions. Thank you.
Okay, over to Richard.
Yeah, hi, Beth. I saw two questions for you.
Okay.
Let's see, the first question is Annex 1 nowadays. Is the industry referring to the Annex rewrite or still refer to 2007?
Oh, that's a very good question, because you should be following 2007, but you should start my video. Oh, did I come out of video?
Yeah.
There I am.
Right.
Okay. So you should be looking at what's coming in order to prepare for it, because once it hits, there's going to be a little bit of time, but you really need to start moving in that direction. So again, where there's issues that you have enough data to push back on and you don't want to do that, for example, you don't feel like dedicated socks are a thing for your facility, then you need to make sure that you have a program in place, maybe where you have dedicated shoes and you're doing something else, or you're decontaminating your shoes or something. But you should be looking at the new draft and getting ready for what's coming, because I mean, even from 2019, when I was in Europe, I heard everybody talking about PUPSIT, this pre-use integrity test. So you should already, realistically, you should have already been doing that. But the Annex 1 now is saying, you need to do that. So some people may have not been doing it. So you really need to be looking at the new document and moving in that direction, but take a look at what's there and see if it makes sense for your site. And then just kind of be involved with that process and know what people are contesting and what the dialogue is happening in industry, because you're going to see, okay, this is a hot topic right here. This is a point of contention. I hear a lot of people talking about it and they're arguing about it. Well, that clause may change. But if it's something where nobody's really talking about and you're not doing it, then you need to do it.
Okay. So the second question is it advisable to have a per product cleaning validation or perform grouping bracketing of products for cleaning validation?
Read that again, because it cut out in the middle. Sorry.
Yeah. Is it advisable to have a per product cleaning validation or perform grouping and bracketing of products for cleaning validation?
Oh, per product. Okay. No, I would group. It's going to save you time. It's risk-based. It makes sense. Where people get in trouble is they don't document the thought process. They don't do their testing. They're picking products that may not be worst case. Just by looking at solubility tables, you need to consider everything. You need to do a risk assessment, but group, bracket, definitely. But remember, when you're doing that, those products, they need to be cleaned by the same process. The equipment needs to be similar. So there's other things that you need to look at rather than, just these products. But I would group your equipment, I would group your products, I would matrix everything out and have a plan in place and document.
Yeah, I think that's all. I didn't see
That's
any
it.
more
Well, that's easy.
questions. Yeah, no more questions.
Great.
Chat box.
I'm afraid you're not off the hook. There is a question.
Okay, that's fine. Well, and I always have my email. People have this presentation. They can contact me if you have questions after. So yeah, if there's another question, I'll be happy to take it.
But it's got acronyms in. But before I ask that question, I'd like to remind everyone if they could please help us with feedback. There's a link that's been posted on the chat. If you could just click on that link, it will take you to a form that you can just fill in and it will come back to us. So I would appreciate that. So the question from OBK is, when chemical does not have NOAEL and LOAEL, can we use NOEL as point of departure to calculate ADE? If we use TTC, it may be stringent. In PDA TR29 and APIC CV guideline, LD50 is linked to NOEL. Is this relationship correct? LD50 is not a means to derive ADE.
No, LD50 is not a means to derive ADE, but you can use null. There is a modifying factor on the PDE that allows for that, so you can do that. And I recommend getting a toxicologist involved for something like that if you're going to start using a different point of departure.
Thank
to
you.
justify that. So yeah, and your cleaning agents, you know, I don't recommend looking at the individual cleaning agents and coming up with your own, you know, look at the formulation. I know at Steris, we provide HBELs for all of our products. So if you need this data, just reach out to us. We have it for, our top products that are, I'm not top products, we have for all products, but if you're using, one of our flagship products, then by all means contact us and we have that data.
Thank you. There's a question from John Lim. If final conductivity fails at the end of CIP, do we need to perform revalidate the CIP validation?
Okay, so if final conductivity fails at the end of CIP, do you mean during validation or do you mean in routine use? Because final conductivity, and then is it failing every time? Is it something that's, systemic, or is it just a one-off, like somebody, stepped on a line, or your conductivity, an air bubble, or, a conductivity probe failure, or, whatever, transmitter connection. So there could be a lot of things. So if it's just a one-off and your conductivity fails one-off, what I would do is I would obviously, you need to investigate and see what's going on. Perform your cleaning again because something happened. And if your conductivity is off, investigate that and why it's off. But if it has drifted to a point where it's continually failing, then yes, you need to see what's going on with your process. And maybe there was a change that you didn't know about, such as, a new raw material that's harder to clean, that's kind of dwindling and coming off and coming off in your rinse and not coming off adequately in your wash step. So yeah, you could have drifted out of control and then you need to investigate it. Do you need to revalidate it? I wouldn't jump right to revalidation because you need to find out what the problem is first. So, if it was a minor change, obviously it's enough to impact that conductivity if it's continual. So you may have to, yes.
John, does that answer your question?
Yes, thank you very much. I'm outside, so I just
Okay.
Was it a one-off, or I guess we can take that offline if you want. If you can contact me by my email if you want.
Okay, I think that was the final question. Unless there are any others,
Okay.
this is your golden opportunity to unmute or type in a question on the chat. Otherwise, please do click on the link that I've just sent to everybody. That way we would appreciate your feedback for future sessions. Okay, so someone's just taken over the screen sharing. Sorry.
That's okay. That's a beautiful picture.
Yeah, Michelle is sharing a picture of Switzerland, it seems. All right, so thank you very much, Beth, for your time and
no problem.
Us. And we look forward to future opportunities to welcome you back. So without,
All right, thank you very much for the time.
thank you very much.